Hello again. This week's pre-lab lecture is covering material you will need to know for our fourth week of RT-PCR. So this is the fourth week working in section 1, utilizing the assay reverse transcriptase PCR. Today we are going to be setting up our reactions for the actual polymerase chain reaction component of RT-PCR. So just a few notes about this week's procedure and in-lab activity. So if you've read through the protocol, you will have seen this before, but I'm making a note of it here. Your instructor and or your TAs will provide you with your TBP master mix, right? So if you remember, TBP is our housekeeping gene that will be comparing gene expression of all of our other genes of interest to. You'll be handed a solution that is basically ready to be mixed with your treatment samples right out the gates. So you don't need to worry about making this. You will be given it, so make sure you know where it is and you don't confuse it with your other reagents. A note about the protocol and some of the reagents you'll be using today. The PCR reagent is the green solution on your benches. Please don't confuse that with other reagents that you will be provided or solutions you'll be making. And just a quick note about the inclusion of a no cDNA control with our experiments this week. So in some labs, or most labs anyway, when using RT-PCR for, I'm air quoting, in real world experiments, groups will oftentimes include a no cDNA control as well as a no reverse transcriptase control. So in a no cDNA control, no mRNA sample is included with the reverse transcriptase reactions, right? So if there's no mRNA included after reverse transcriptase does its job during that reaction, there should be no cDNA produced. As opposed to a no reverse transcriptase control, in these samples, your mRNA samples are loaded into your tubes. However, we do not add or include a reverse transcriptase enzyme. So even though there's mRNA present, there's no enzyme available to convert your mRNA to cDNA. So both of these are meant to simply control for contaminating genomic DNA, but for our purposes in lab, a no cDNA control will be adequate. Again, just to reemphasize, some or most labs will use both controls to confirm various things about their samples. Today, so what this does mean is today you will have 4 samples or reactions per gene. So the no cDNA sample is now an extra control in addition to your control treatment sample. So in total, we will have a no cDNA sample, a control sample that received, this is when our zebrafish received no drug treatment or toxin treatment, and then our drug replicate 1 and our drug replicate 2. That totals 4 samples or 4 reactions per gene. And this will mean the total number of solutions you make today depends on how many genes you have, which depends on how many students in your group you have. So notes about the cDNA samples that would be provided to you if they're not already on your benches. So between last week and this week, your instructor has converted our RNA samples into cDNA. So to do this, your instructor has used a reverse transcriptase kit. So this kit combines your RNA samples with a solution that contains reverse transcriptase, as well as RNase inhibitors, because we're still trying to avoid unwanted degradation of our RNA isolated from our samples. It will also include excess nucleotides, which reverse transcriptase is going to use to convert or copy our RNA and make a DNA strand. It will also contain those oligo DT primers, right? So these DT primers are what allows us to selectively only reverse transcribe our mRNA to DNA, because this will help us select out for that poly A tail found in mRNA. And then it will also include other random primers to help promote reverse transcription of all of our RNA. So this note about this reaction, it only takes 1 cycle to produce 1 copy of RNA to DNA. So it's a fairly short process in the thermocycler. It's just one episode of heating and cooling, as well as some denaturization steps. Anyway. And I have previously provided specific information about this kit, including the protocol for you. If you're having trouble locating that, just simply ask, or if you consult your protocol, there should be a link or the name of this kit, and you can look it up on your own. So this reverse transcriptase event, again, produces a one-to-one copy of all of our mRNA transcripts to cDNA. So for every one mRNA transcript present, you will now have one cDNA transcript present. And so what we're going to utilize that today is if there was 10 mRNA transcripts for a certain gene, right? All 10 will be transcribed into cDNA, and now we're going to amplify those products. So what will eventually come out of this is that a gene where only one mRNA copy was present at the start will amplify 1 to 2, 2 to 4, 4 to 8, 8 to 16, whereas cDNA, a transcript that had 10 copies will amplify at the rate of 10 to 20, 20 to 40, 40 to 80, right? They both double. It just depends on how quickly they double, just depends on how many original transcripts there were. So, last note, your cDNA sample contains complementary copies of DNA to all, yes, so this is exactly what just happened, right? Our cDNA are just complementary copies of our original mRNA transcripts. Oop, and I was supposed to load this animation before. So here are our oligo DT primers, right? Again, they're going to select out for that poly A tail. They're going to prime that reverse transcriptase enzyme and allow that mRNA single or 10 copies of mRNA to be transcribed into cDNA. So what you're making today are stock solutions and master mixes, right? So the first solution you're going to be making is called a primer mix. And this primer mix contains the specific primer sequences that we previously generated weeks ago, right? When we did our NCBI webpage activity where we picked our genes and we went through the process of developing primer sequences, and we wrote down annealing temperatures and amplicon sizes. The reagent you guys helped build and construct is what's going into that first solution. So these primers, specific for each gene, allow us to selectively prime and amplify only our genes of interest in the solutions that contain our primers, right? So just a little animation here again. So these primers that are specific to each gene of interest, so we have a forward or reverse primer sequence here, allows us to selectively amplify only our genes of interest, right? These primers won't bind other genes, only our genes of interest. You will be making one primer mix per gene of interest. So if you have 4 genes of interest, you will make 4 separate primer mixes, right? And all these primer mixes will contain both the forward and the reverse primer sequence that you developed. So the last note about this, if you have 4 genes of interest, recall that we also have to include our housekeeper gene. So technically our TA and your instructor will be making that one for you, but in total we would have 4 to 5 primer mixes, including our housekeeper gene. But you will only make primer mixes for your genes of interest. So the next solution is called our master mix. This contains PCR reagents, so things like nucleotides to help the reactions move forward, to basically give fuel to our DNA polymerase, which is this guy right here, right? TAC polymerase is our special heat-resistant polymerase. Heat-resistant DNA polymerase that we're going to be using this week. So our PCR reagents contain those nucleotides for fuel and that special DNA polymerase. And in addition to that, you must also include a small volume of your primer mix, right? So that's why you have to make your primer mix first, because the primer mix is a component of your master mix, right? This will give our reactions the supplies they need to amplify only our genes of interest. And then just to reemphasize, so our master mixes will now contain again, that small portion of our primer mix and our DNA polymerase. And so every time we cycle through the thermocycler, that DNA polymerase will utilize those primers and amplify only our genes of interest, doubling each cycle. So this gives our supplies, or gives our reactions supplies they need to amplify. You will need 1 volume of master mix solution for each sample. So when doing your calculations, you should see in one of our tables that we give you the math for the volume necessary to complete 1 reaction with 1 sample. So in order to generate enough master mix to use for all samples, there's some multiplication. You should be able to follow this in the table in your protocol. So again, as a reminder, we will have 4 samples, including our no cDNA control. So that's no cDNA, our control, right, the group of larvae that received no toxin, and then our drug replicate 1 and our drug replicate 2. So you will need 4 total volumes of master mix, but I believe our protocol is going to have you make 5 volumes of master mix just in case there are small pipetting errors. So you have plenty of reagents to work through today. So again, as a reminder, you will be making all of these stock solutions for all of your reactions except for making the master mix of our TBP, or our housekeeping gene. Your instructor or TA will make that for you and provide it for you. You need to make the rest of the primer and master mixes for your genes. Again, PCR reagent is green. Don't lose it. Don't confuse it with something else. Should be labeled. And then a note about organization. So today you will be working with anywhere between 12 and possibly 20 different small tubes. I recommend leaving tubes in strips that you can, right? So strips of 4 would allow you to have one gene for each sample. So if you leave everything in a strip of 4, you can have the first tube as your no cDNA control, the second tube your control, and then drug replicate 1 and drug replicate 2, right? If you leave things in strips of 4, you won't confuse genes of interest across other genes of interest. And this will hopefully make more sense when you see the diagram on the next slide, as well as once you start reading through the protocol and actively doing this exercise. And the last note, so remember those annealing temperatures that we required you to write down? We require this because each different primer mix or primer sequence will have a unique annealing temperature, and we essentially have to set up a different thermocycler program for different annealing temperature preferences in our sequences. So one of our thermocyclers will utilize primer sequences that had 52 or 55 as our annealing temperature. This guy is going to be on the left if you're facing the back of the room. And then the thermocycler on the right will be used for primer sequences that had annealing temperatures near 60. If you have any questions about this, please feel free to ask, but your instructor and your TAs will help organize this and make sure every reaction goes in the place it needs to. So this diagram is meant to help hopefully avoid some confusion. It's meant to basically represent everything you'll be doing today, right? So over here on the left, this, and just as a note, this whole diagram is built as if a group of 4 was performing today's lab. So over here on the left, we have our primer mixes, right? You're going to be making these up in very small volumes, so be careful when you're pipetting. We have a primer mix for our housekeeping gene. Again, your TA or instructor will make this for you. However, we also have a primer mix for gene 1, whichever you chose, primer mix for gene 2, gene 3, gene 4, etc. You use your primer mix as a component of your master mix. And again, you have 1 master mix for each gene, right? And again, noting your instructor and or TA will make the master mix for TBP. You will make a master mix for gene 1, master mix for gene 2, etc. So 1 master mix for each gene, including TBP. You will use a very small portion of that master mix to set up a reaction with each of your samples. So in this example, you would take 15 microliters of your master mix for TBP, put it into a tube, and then you will take 5 microliters of your no cDNA control and also add it to this tube. Now you have a tube that should have no cDNA in it, but the master mix for TBP. Let's move on to this example here. So again, 15 microliters of master mix goes into a separate tube. Now 5 microliters of the cDNA from your control sample will also go in this tube. That means that we will only amplify our gene TBP in this control sample. Yep, and so rinse and repeat. You'll make one of these tubes for your drug replicate 1, and you'll make a tube for drug replicate 2. So what will occur in the thermocycler is in all these tubes, only TBP will be amplified, and when we're all done, you can compare how much TBP was amplified in your control versus how much TBP was amplified in drug 1 versus drug 2. And the same will happen for every single gene setup and every single control. So this is what I meant when I said keep your tubes in strips of 4, right? So you can label this whole strip as TBP and then just label no cDNA, control, drug replicate 1, drug replicate 2. That will help you keep your TBP gene tubes separate from, say, your gene 4 tubes. If you have any questions about this, please feel free to ask. Hopefully these arrows help. And again, if you have any questions, ask before you start pipetting. This is a representative figure of what the inside of our thermocycler layout looks like. It looks very much like the 96 well plates we've used in class before. So when we ultimately put our tubes or reactions into our thermocyclers, if you have already left for the day, your instructor will load your samples into the thermocycler and take them out exactly as they went in. So if you fail to label your tubes in an effective, clear manner, this will be the error on your group's part, right? Be sure you are labeling things clearly. I make this note because once I put the tubes into the thermocycler, the reaction can take several hours. Sometimes it runs overnight. Your instructor will not necessarily remember whose samples went where. So you need to label them clearly with your lab day, your group number, your sample ID, and the gene of interest. You need to label these clearly. Again, your TAs or your instructor will help verify your samples go in the correct cycler. We have that information. If you want to stick around after you finish to make sure that occurs, feel free to do so. And a comment and a reminder to use your pipettes correctly. Today we are pipetting extremely small volumes of substances, right? If you look closely, the amount of primer mix or primer solutions is very, very small. You need to pipette carefully and precisely. Another reminder to only use filtered tips. You will be provided with these. This means you should not be using tips from within your drawers. The tips within your drawers are not filtered. Your instructor has plenty of filtered tips and you will be provided with them. Please do not use the incorrect tips. If you have any questions, please feel free to email me.